My name is Kathryn Atherton, and I am presenting my poster, The Interactogram, Understanding the Biology Behind Association Networks. Urbanization is rapidly increasing worldwide, but the impacts of urbanization on the environmental microbiome are poorly understood. In Massachusetts, soil microbiome association networks disintegrate with urbanization, as seen in the box plot. Association networks created from microbial abundance data are often used to infer ecological processes like species interactions. Yet, these networks can reflect other community attributes, such as richness, diversity, and spatial autocorrelation, as well as similar responses of different microbial taxa to the same environmental stressors. An example is shown below the box plot. Microbes that are positively associating due to a cross-feeding interaction will be observed to have the same co-occurrence pattern as microbes that are positively associating with an environmental variable that the network is blind to. This has impaired our ability to assign ecological meaning to microbial networks, a critical issue for systems where networks may reflect important microbial processes that impact ecosystem function. As such, the goal of this project was to layer biology onto the network statistical methods to understand the types of associations, shared environmental responses, competitive exclusion, combat, and cross-feeding in networks. And we hypothesized that different genetic pathways would be enriched in each type of association. Stress response pathways such as heat shock proteins or xenobiotic degradation might be indicative of a shared environmental response. Resource decomposition pathways such as cellulose and carbohydrate degradation might be indicative of cross-feeding interactions. Nutrient metabolism pathways such as the central carbon metabolism or nitrate reduction might be indicative of competitive exclusion, and antibiotics production or resistance might be indicative of combative interactions. Thus, we developed the interactogram to categorize these associations based on the network edge weights and the metabolic similarities of associating microbes. After constructing co-occurrence networks of bacteria and fungi at the edge and interior of urban and rural Massachusetts forests, we extracted the edge weights or association weights of the microbes. This value is plotted along the x-axis of our interactogram, as seen on the bottom left figure. We then calculated the Jacquard similarity of the associating pairs from enzyme profiles, which we used as our metabolic similarity value on the y-axis of our interactogram. We set the threshold of high and low metabolic similarity at a Jacquard similarity value of 0.6, as this is the average Jacquard similarity between different microbial functional groups in our networks. Pairs of microbes that are positively associated and metabolically similar are expected to be associated due to environmental preferences. Positively associated and metabolically different pairs are expected to be cross-feeding. Metabolically similar and negatively associated pairs are expected to be competing for resources, and pairs of microbes that are metabolically different and negatively associated are expected to be engaging in combative associations. We then used an ANOVA analysis to determine which quadrants our pathways of interest are enriched in. Our resulting interactograms can be found on the right. The urban forest network results are on the top, and the rural forest network results are on the bottom. Forest edge networks are on the left, and forest interior networks are on the right. The colors of the points in the interactograms represent the pairs of functional groups that are associating, and the sizes of the points represent the number of mutual edges. The colored ellipses represent the pathways that are enriched in one or more quadrants and are colored by the pathway type. The exact pathways enriched in each quadrant are listed and colored by pathway type. Asterisks next to the pathway names represent the significance level. We found that the xenobiotics degradation pathway, represented by the green ellipse, was enriched in both the shared environmental response and competitive exclusion quadrants in only the urban interior network, suggesting that xenobiotics are both a stressor and a resource for urban microbes. Decomposition pathways, represented by blue ellipses, had different enrichment patterns across the networks, but were predominantly found in the competitive exclusion and cross-feeding quadrants. Nutrient metabolism pathways, represented by pink ellipses, were enriched in competitive exclusion, combat, and cross-feeding quadrants in the urban and edge networks, suggesting that the heat and excess nitrogen in urban forests and in forest edges select for faster growing microbes. Antibiotics production and resistance pathways were equally enriched in combat and cross-feeding quadrants across all network types, suggesting that antibiotics can be a strategy for both offensive combat as well as potentially a commensal protection mechanism. In the future, we will aim to provide confidence estimates for the likelihood of associated pairs assigned interactogram interaction type. Thanks so much to the Boston University Urban Program in Biogeoscience and Environmental Health and NSF-NRT, as well as the Boston University Microbiome Initiative for funding, and to David Heaton for his help on this project.